Hello, AP Bio. I'm going to quickly go over the slides that I wanted you to take notes on. We talked about the end of class about the actors being the parts of photosynthesis. Now, we have the two cycles, one being the light-dependent reaction where electrons are being excited and then reduced in terms of their energy, which is powering the production of ATP and NADPH. And we are using light through a series of pigment transferred proteins that absorb electromagnetic radiation to excite those electrons and to oxidize water. In many ways, this is different and parallel or opposite to the ETS in cellular respiration, where we are forming water from oxygen based on proteins having electrons be reduced by NADPH being oxidized. So it's the opposite. Water is being oxidized rather than NADP, and oxygen is being formed rather than consumed. And then the second cycle is where we are going to be adding the ATP and NADPH energy by oxidizing them to reduce glucose molecules or sugar molecules using an input of CO2 rather than reducing sugar to form NADP and ATP as well as forming CO2. Lots of obviously proper nouns. The key difference is being whether it's reduced or oxidized. So the photosystems are two complexes in the thylakoid membrane. The thylakoid is part of chloroplasts and mesophyll cells. So chloroplasts being an organelle, and within them there are stacks of these membrane things called thylakoids. Stacks of thylakoids are known as granum. And outside of the thylakoids is this fluid called a stroma. So this right here is the chloroplast, and then here is a thylakoid within it. And we have two systems. We have photosystem 1 and photosystem 2, which use P680 and P700 as pigment proteins that absorb light. And that light energy is going to be transferred between different pigments within these photosystems until P680 or P700 absorbs that light energy and excites electrons. And all of these accessory pigments are used to transfer energy from a relatively high electromagnetic radiation wavelength, such as 300 or 400 nanometers, to the 680 or 700. And that is going to reduce excess heat, because if we go from 700 nanometers, or I should say 400 nanometers, a high energy wavelength, to 680, there's going to be a big drop in energy, so heat would be produced, and that would often lead to the death of cells if all of that heat was generated. So light energy sort of passes between each pigment, sort of like a pinball machine, until it becomes concentrated at the reaction center. And this is sort of the idea of funneling the light into the chlorophyll reaction centers. Now, light is the energy driving the creation of chemical energy in this cycle, and light has energy based on the wavelength of electromagnetic radiation. So we are talking about wavelengths of energy from anywhere between 400 and 700 nanometers, where smaller wavelengths means that the energy is hitting a surface or an object more frequently than a longer wavelength, because frequency is the inverse relationship of a wavelength. So a larger wavelength means that fewer waves hit the object per second. And a smaller wavelength means that an object is hit by photons of that energy intensity more frequently. More energy, though, means the capacity for more work, but also it means the capacity for larger heat production if we use that energy without a mechanism to control it. So pigment proteins and photosystems absorb different wavelengths of visible light, whether it is high or low, and they can use that to excite or reduce the electrons in P680 or P700. The absorption spectra of each pigment shows which wavelengths of light energy that pigment can absorb or reflect. So when I'm looking at chlorophyll A Chlorophyll A is really good at absorbing energy light from about 400 to 450 nanometers. Chlorophyll B sort of is peak absorption between 450 and 480. And carotenoids, which we often see in carrots, are a little bit lower in terms of energy absorption. And then from about 500 to 600 nanometers, none of these pigments can really absorb or use light. And then we see a large peak at 680, 650, and 700 for chlorophyll A and B. And this creates an action spectra, which basically shows what is the relative amount of usable light for photosynthesis based on a plant with these three pigments. So we could use a lot of high energy and a lot of low energy light to excite electrons. The accessory pigments in photosystems, carotenoids being one, not only allow plants to absorb a variety of wavelengths of light, but also it helps reduce the energy jumps. If we were to simply take 400 nanometer wavelengths of light and use it to boost the P680 or P700, that'd be a very large drop in energy, which would produce a lot of heat, causing distress to plants. And here are some of the different pigments that we can see. So we have chlorophyll A and B, phyrocerinin and phycocyanin are two pigments that evolved in algae before plants evolved. So the reason why many plants fixate on chlorophyll A and B is that when plants were evolving in an oxygenated atmosphere, the algae already were able to capture sunlight of these intensities. And what plants are doing with these accessory pigments is absorbing the light and then using accessory pigments to transfer the energy to a lower state to then excite electrons that we get from the electron donor. In this case, the electron donor is going to be water that is oxidized. So let's put it together. Both photosystem 2 and photosystem 1, 680 and 700, want to use electromagnetic radiation at lower energies to excite electrons. The movement of electrons through the ETS is going to drive chemiosmosis of hydrogen ions, and it's also going to help with the reduction of NADPH. With the absorption spectra of chlorophyll A and B plus accessory pigments, light is going to be transferred from one pigment to another until they reach the P680 and P700 pigments, who use energy intensities at the lower end of the visible spectrum to excite those electrons. When that energy is absorbed and transferred from light into P680, P680 is going to reduce a pair of electrons or excite them, and those excited electrons will flow through a protein system. So we're going to be seeing the movement of electrons through each of these pigments from high to low, 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 meaning that each of these pigments has a slightly higher and slightly lower energy intensity that they absorb light energy at. And once we excite those electrons, they are going to flow through different protons or proteins that are more electronegative than each other, which will help drive the chemiosmosis or the concentration gradient of hydrogen ions, which will flow through ATP synthase to produce ATP by reducing or phosphorylating ADP. The very electronegative P680 pigments that are energized will then be oxidizing a water molecule or stealing electrons from H2O, because P680 as a molecule is a more electronegative than H2O. So we are going to be reducing oxygen or making oxygen lose electrons, and those electrons will be used again to start the process of electron excitement via light. The hydrogen ions are obviously going to help form a concentration gradient, and we get a lot of those hydrogen ions by splitting water molecules, which will want to flow out of the thylakoid membrane into the stroma or the chloroplast innards due to a concentration gradient. For now, I think that is enough information for photosynthesis. the main ideas that I'd like you to feel comfortable with is that we use electron excitement and reduction to power a hydrogen concentration, which power ATP. We also use the same energy to reduce NADP into NADPH. The only way we can excite electrons is by concentrating light energy through a series of energy absorption reactions using different pigments that are going to be taking high energy electromagnetic radiation, such as blue light, 400, 500 nanometers, and concentrating it into 680 or 700 nanometers of wavelength. And that those different accessory pigments absorb different frequencies of light based on their chemical structure. Have a great day, and I will see you soon.